Our very first task is to thank all of you, thank the branch, for sending us to Portland to this Congress. It was a remarkable experience to go through, which we'll talk to you about. And so we are both very, very grateful indeed for that, for being sent over there. And we hope to deliver to you something really interesting as a result. Clearly, it won't be deeply scientific, although there's been plenty of scientific information for those who are able to absorb it. So, let's go first of all to the opening slide. There were 4,500 delegates at the Congress. That's quite a bewildering number of people to encounter all in one place. But as was pointed out by John Strussel, the convener, there's a proverb in Africa that it takes a village to rear a child. And I reckon 4,500 people is about the population of a village. But his idea was, we are a village, and we can take care of Parkinson's.
Yes, he said that it will take a village to beat Parkinson's, and we are a village. It was very impressive. Yeah.
So, yes. Are we going to dim the lights a bit further?
Sorry.
Thanks, Helen. Okay, just makes it'll make the slides a little bit easier to see. Okay.
Good.
Yes. And you can see that picture in the auditorium. That's 3,500 seats. This is the entrance place where everybody was registering, and this is the poster place. The whole thing is quite enormous. Now, does that take us to the next one? So our journey began with this lovely mountain, Mount Hood, which overlooks Portland. The city of Portland itself is a lovely liberal place. There's a great atmosphere there, and we felt quite at home there.
Yes, it's almost like the same sort of size as Edinburgh. You can walk everywhere. It's just wonderful.
Yeah.
So.
And the rivers, there are two rivers that bound it, the Columbia River on the north and the Willamette, and you can see that's the Willamette River, beside which, and in you can just see there, that's the Congress, the conference center. A flood of impressions. One of them was people walking around saying, "Keep Portland weird," which is good.
Well, but we did our best.
And as you get on the transit from the airport, there's this little voice that says, the train's departing, the doors are closing, please hold on. And this was a kind of metaphor for the whole thing, because the Congress went at such a speed that we had to hold on to our hats to keep up with things. So advancing science, promoting community, and inspiring hope. Those were the objectives of the Congress, and I think it really achieved all of those things. We met some astonishing people. Tom Isaacs.
Tom Isaacs very much dominated the Congress. He's been diagnosed for 20-odd years. I think he got his diagnosis at the age of 27. And he's the one who co-founded Cure Parkinson's, which is a very powerful research and fundraising thing in Britain. But he's not frightened to speak his mind, and because he's so well regarded, he was able to say things that other people weren't able to say. So there's a few of those things will come up later on. And he also has written an amazing book called Shake Before Use. So he referred to himself in the very beginning as one of the shaky people. So uses humor a lot to deal with things.
One of his remarks was, "Witch doctors are witch doctors." David Leventhal, who a number of you will have heard or even met because he came over to start the Dance for Parkinson's classes with Scottish Ballet. He also, these two both got the World Parkinson Congress Award for contribution to the Parkinson community. Very, very impressive man, and trained with Mark Morris. And what was his line?
His line was that he wants to see people with Parkinson's moving with grace, passion, and power. And I just want to pick you up. He didn't actually. He didn't start the Edinburgh one. That started a long time ago.
Yeah, well, he started the Scottish Ballet one.
Yes, he came over to train the Scottish Ballet one. Just you know, I've been told to pick up small points.
I have to be corrected on these things.
And I had a wonderful. I got invited to one of the training sessions that I think some of you remember. Virginia Belt, who came over and danced with us, and she invited me to one of the sessions. So I had a session with David Leventhal, and it was just wonderful. So.
Dawn May.
Dawn May, she lives down in Bristol, and she's written a series of books, and I've got a couple here at a tenner each, called Does It Hurt, Granny? And it's books of how to talk to grandchildren about your Parkinson's. And in reading it through, I thought this is wonderful, so I've got ones for all my grandchildren for Christmas. And she's actually she's talking to the people who did the Wallace and Gromit films, and she's probably going to animate them, which is lovely. And Gaynor Edwards, she's a stunning young woman. She's been diagnosed for 4 years. She's in her early 30s, and she runs something called Young Onset Parkinson's Spotlight. She's one of these very powerful, very scary ladies, and I thought she was keeping Portland weird as well.
Yeah. So we really met, and that was just the tip of a very large iceberg of amazing and brilliant people. The things we found were the highlights for us were the capacity of everybody to listen. All of the presenters were really alert to their audiences and listened to people with Parkinson's and their questions and responded with great insight and respect. The other thing was that we should take control of. I worry about this word control, but it's one of the ones that Parkinson's UK have highlighted, and it's something certainly to work on. But it means that the initiative of the person with Parkinson's really, really matters, and of carers too, of course.
Yes, and the Congress had at least a third of the people there were people with Parkinson's and their care partners. So we were very well represented, and our voices were very loud.
And then the power of community, which was palpable in the place. It really was a wonderful feeling of being among people who were all in this together.
Yes.
So, what are we doing personally as a result of this? Well, more action and responsibility for our own well-being, and that's both of us. That's both of us, and more fun.
Yes, so the emphasis on the whole Congress was about fun and humour and getting involved and participating. And we did, I mean, it was hard work. It was huge fun. It was wonderful. So again, thank you all for sending us.
And the hard work continued in putting this presentation together, I can tell you. But we've made that fun as well. So here we are back into the Congress itself. It was enormous, quite overwhelming. You've got lots of statistics up there, but we did manage to attend about 40 presentations each, or 30 or 40 presentations each. And that was exhausting. A little note at the bottom here, feeling weird, going to chill out in the meditation lounge, kind of see you soon.
Yes, I wrote that halfway through it, flaking out halfway through a session, and just went into the meditation lounge and slept. And the program here is so big and so heavy that I had to just tear mine out and take one page at a time in with me. I couldn't carry this around with me the whole time. So we'll leave it there if anyone wants to have a look at it at lunchtime.
But being a beast of burden, I hung on to mine, so bring it home, show you all. But it was astonishing. I mean, the parallel sessions and everything else, you couldn't really, to find one's way through was a big challenge. Anyway, got my certificate, got the T-shirt. Here is Parky.
This is the miniature version. That's the
and you can see the big version there. There's Alison doing a talk for Care Parkinson's. We're collecting ideas from there. But you can see that the big Parky himself is already cross-eyed because he's under the weight of information that he's carrying, responsibilities he's carrying. So. There's something there. Yeah, a few caveats. I'll have to look at the screen here. The stories are just from the sessions we managed to get to. We couldn't cover the whole conference. Thilo is going to come later. Thilo Kunat will be with us later on, and he's covering, he's got some reports from other areas of the conference, so you'll get quite a lot of interesting things from him. We came away with many more questions than we arrived with. We can give you links to anything you particularly want to follow up, because obviously this presentation will be pretty superficial, I'm afraid to say. It covers a lot of ground, but it can't go to depth.
Now, yes, this is the big caveat.
And then we our imperfections will become manifest before you.
On your tables, you've each got either a cush ball or a brain. And the idea is that if we go on too long, then throw them. And we've got a brain as well. Some of the brains are bright orange Fox Foundation ones, and some of them are rather ancient and crumbly grey ones. So, whoops.
So if and when you've had enough, throw something.
So I shall sit at the side with something to throw and leave you to it.
Yes, I would ask you to ask a question or two first, of course, but that's another. This is obviously the story beginning. Once upon a time, there was James Parkinson wrote this tract, essay on the shaking palsy, which is the beginning, and that's 200 years ago. And where have we got to since? You heard the story of the blind men encountering an elephant, and one says, well, this is a very sinuous, sort of snaky kind of animal, and the other one says, it's got a strange sort of bushy thing. Is that a tail? And the others say, well, no, actually, it's a tree. He's got, and so on. And they couldn't agree what this animal was, but they did discover eventually that it's an elephant. And we're wondering if after 200 years, are we now really seeing the elephant? Parkinson's affects people in many, many different ways and makes no distinctions as to the kind of people it infects. This is from Canada. This is a drag queen who's put herself wonderfully forward as saying, we're all in this together. So we thought that was entertaining. But this is the background perspective. Obviously, Parkinson's gathers, as we know, a long time before it actually shows up in something that people can diagnose, and then has all these different, both motor and non-motor manifestations. So that's the kind of the fundamental that we're looking at. We're also looking at a picture where it increases according to age, although there's maybe quite a lot of perhaps neglect of people, younger people with Parkinson's, that clearly it does build up in an age profile. And it has a worldwide impact, something like 5 million people anticipated with Parkinson's by 2030, I think that says, and with a major economic impact. So that was just part of the scene setting. And here's what we have taken from this. So there's a lot to do with the science in terms of understanding PD and what is being done about the different motor symptoms and non-motor symptoms. There was a lot about non-motor symptoms there, which is good to see. And then the business of taking control, body, mind, and partnership. What does that actually mean? And then the role of community, which came forward very, very strongly, because we all, nobody should do this alone. So our review covers quite a daunting range of stuff. A look at levodopa, a mention of DBS, a mention of alpha-synuclein, because you can't ever get anywhere without covering that to some small degree. Quite a lot about the work on cells and restoration, potential for restoration, something about genetics, and then something about disease modeling that's coming forward. This was the lecture, the special lecture delivered by Professor John Nutt about on where do we stand with levodopa. He presented really the unique features, which it does have a very short half-life. It's absorbed from the small bowel, and the stomach is actually destroys it if it doesn't go in the right packaging, so to speak. And all the complexity of getting levodopa into where it then can act. And it's a prodrug, so it has to be changed into dopamine and so on in the whole brain system. Problem is that this very short life of levodopa in the blood, and if I go, and then the whole business of how do you manage absorption. Immensely complex, and I think this is what the Congress taught me, was the extraordinary subtlety and complexity of the human body and its response to different medications, and how the skill and almost skullduggery of getting medicines into the right places in the human so they can do their work. And so that just shows some of the complexities in the process. And here's another extended explanation of what happens. So we've got the esophagus, the stomach, the intestine, the liver, the heart, the lungs, the heart again, and the brain. And that's the passageway, if you like, so that only 2 or 3% of levodopa actually reaches the brain. I hadn't realized when I went there that was such a tiny amount that actually, of the dose that actually reached the target site, if you like. So, and so there's a colossal effort going into how, by what mechanisms can this be improved, extended? How could you devise some continuous process to refresh the levodopa? And then a lot of information about the gut. And I just put this slide up because it was it showed to me something about the intelligence of the gut that we have, and how clever and subtle it is. And the amount of science needed to actually get through this kind of process. So that's just an illustration. But I thought it may look a bit gruesome, but there's also quite a lot of beauty to it. But what John Nutt came out with was that obviously levodopa will remain a priority in terms of therapy. That research needs to focus on more than continuous delivery and consider variable delivery in relation to different patients' needs. And that's really complex. And new methods that measure motor function can help with feedback into this. So there's a huge growth of technology about this.
I was just thinking, Would it be worth going back to the slide which was looking at the different methods of delivery that were being looked at? Could we go back?
Yeah, happy to go back. I'm appreciating...
There we are, that's right. Because that was one of the talks that I was at.
Yeah.
And they were looking at different ways of getting it past the stomach and the liver. And so talking about infusions into the upper small intestine. And over lunch, there's going to be a video of a young woman who's actually doing her own infusions into her small intestine. They're looking at different ways of actually producing the pills themselves so that they're protected against the stomach and don't actually start to release the levodopa till they get into the small intestine. They're also looking at patches and belt pumps. I think some people have got patches here. Yes. And the other thing they were looking at was a little thing that sticks up behind your teeth that tucks in, almost like, where you used to put your chewing gum up when you didn't want anyone to see you had it. So they're looking at that as well. And also an inhaler, so that if you have an emergency, you can use an inhaler of levodopa, which would kick in about 5 minutes or so, as opposed to the other ones that would kick in later. And in the film that we'll show at lunchtime of the young woman, she was also using one of those nicotine vape inhalers to deal with dyskinesia. So those are the... if you want more information of those, then we can give you the links to it. But that's what they're working on at the moment. Yeah,
I think that draws a very important point, is the amount of innovation that people with Parkinson's themselves are engaging in and creating. So it's not just the specialists, the medics, the neurologists and so on who are dealing with this. It's actually people with Parkinson's themselves who have had scientific training and things like that, but they really are getting into the business of how do we innovate and help ourselves. Okay. So we took a look, we didn't actually, we dodged deep brain stimulation because it was just overwhelming. We couldn't cope with that. Thought, well, actually, this is something I have to leave behind. But I did meet out in the front of the Congress Hall a lady with a hula hoop, and we started chatting. I was with Catherine. Sorry, did I say that Catherine Crawford was intending to come today, and she can't actually get here because she had a puncture out in Fife, and the RAC were going to be 3 hours to getting to her or something like that. So she sends profound apologies. But I was chatting with her, and we went out, and then we saw this lady with her hula hoop, and she blithely said that she had DBS, and so I said, well, how did you get onto this? Oh, my doctor simply said, you've got Parkinson's, you ought to have DBS. So I did. And they put in a spare one in case. And I thought, wow, that's an interesting approach. She was Australian and from Perth in Australia. So there's all sorts of things going on around the world, and people are approaching things differently. But I thought that I sort of felt in the UK that might be a more protracted assessment process to administer something like that. And then a mention of this alpha-synuclein. There were presentations, I couldn't get hold of the slides, and obviously some of the presenters didn't put their slides up to be accessed. So, but there was one very interesting set about how alpha-synuclein folds faultily, and then gets itself through the cellular resistance inside the mitochondria, which it then disrupts, and the cell process breaks down. So that was really a particularly interesting piece of research. It is a major problem, obviously, as to what process might inhibit the the extent of this growth, because these misfolded proteins then infect one other proteins and so on. And as we've seen through other brain diseases, there's a major piece of work to unravel that process. But that is going on, and certainly a lot of progress is being made there. But that's as much as I can say about it. But what has also come across is that alpha-synuclein is not restricted to the brain. It can be found in all sorts of organs around the body. So that's just a reminder of that. And really then getting on to the whole business of how could dopaminergic neurons be restored? Is there some process of doing that? And this leads naturally into the whole exciting business of stem cell work. And there are two forms of stem cells, as some of you may know, but it was new to me. Those that come from embryos, the embryonic stem cells, and those that are induced from people's skin and so on. And that's a different kind of process. The point about stem cells is that once they become stem cells, they acquire a pluripotency, which means they can become anything. That's a huge advantage in one sense, but of course a big challenge in another, because the aim is that they should simply become dopamine cells, and you don't want them to grow into other things. So there's a very careful training process that they go through. I mean, this is all layman's language, and Ken would just be able to describe it much more elegantly than I possibly can. But you might as well hear it in the sort of terms of a novice. There are all kinds of studies going on. There are two major lines of cell development on the upper process going on, and then trials of actual transplants into people of stem cells, embryonic stem cells that go. The problem with the trials is that they've had quite mixed results, some outstandingly successful results and some very unsuccessful results. So it's not at all certain as to how a more accurate process is going. So the research is really important, and we'll come on to how that is being progressed. This is an illustration of embryonic stem cells and how they go. Now, I'm not competent to go into all of that, the process that you see illustrated there, but that is the, if you like, the embryonic stem cell process, grossly simplified, but even so more complex than I can understand. But it does show how the cells can become different, help with the different manifestations in the body, and then there's the business of becoming neurons. So then the induced stem cells, which are taken from skin and offer a slightly, perhaps a more direct process, but at the moment can only be used experimentally for modeling, diagnosis, and drug discovery. But that is very promising because of the productivity of the process. Clearly, the amount of fetal stem cells that are actually available is quite small. Whereas the iPS cells can be created in limitless numbers from simple, taking a sample of skin and taking the cells back to that. What's also emerging is a line of taking patient-specific cells, and that shortens the process a bit, because if you can work from the particular patient and produce something that they can be used solely for them, then there's a shorter process that goes on. The whole business of training stem cells to become little neurons is, I had once one of Thilo's colleagues explaining this, and you know, it was like taking them to school. I mean, they had to learn step by step to be the kinds of cells that you want them to become. And it's quite a nurturing sort of process. It was rather charming, actually, but it was quite wonderful to hear about. So one finishes up with huge respect for the people who are engaged in this and their capacity for wisdom in doing so. Yes, please.
Some of the experimentations appear to be successful and some not.
Yes.
Did you get an impression of how hopeful people were?
Oh, I think if to judge by the... Yes, I mean, two aspects of that. Firstly, the volume of work going into this is absolutely huge. And the amount of research funding that's devoted to it is colossal. So yes, so clearly that is an indicator of hope. And I think if take somebody like John Barker, who was presenting a great deal of this work, but not exclusively. I mean, there were many other people doing so as well. And I think he holds a great, Tom Isaacs used to say, it's hope, not hype. So go for the hope, but not the hype. And there are people who would hype up this process a bit and around the world, and that they wanted to damp that down. But the hope in the process is strong and by all accounts quite sound. That doesn't mean that we can expect miracles tomorrow, because not only has this fundamental research got to be perfected, but the trials have got to be worked through and found out, you know, in what circumstances cell replacement can actually work. And then there's the whole process of validating something for wider use. So we're looking at, I don't know, half or a whole generation probably of work that has to happen in order to bring this forward as an effective therapy. It might be that there might be quicker breakthroughs, but at the moment that seems to be the kind of timescale that people are working at, feasible sort of timescale. So it is highly, it's profoundly interesting, but immensely complex and difficult to grasp. So this is Roger Barker's conclusions. So cell-based therapies in PD have produced mixed results, and because of all these issues, and they have to really refine those in order to produce a therapy, a sound therapy. When it works well, it works very well, and attempts are going to design better trials, and that. I didn't catch that entirely, but it's.
Hang on, I'll take the mic down. One second, sorry, Donna.
I'm interested in when it says when it works well, it works very well. What does that actually mean? Does it mean someone with Parkinson's goes back
Yes.
3 or 4 years in the curve, or
Oh no,
does it mean?
there are people who have. Yes, Ken. Let Ken answer this. It's better.
So just to say, some of the early work on fetal stem cell transplants that was done by Patrick Brundin, who gave the Parkinson's lecture here last year, he has patients who were removed from medication after the transplant. They actually survived. I mean, one of them's recently died after some substantial period of time and didn't go back on medication after the operation. So it's completely successful in the sense that any dependency on levodopa was removed by the transplant. Unfortunately, it is not true of all the patients.
Thanks a lot, Ken. And I came across one, he was a South African man, and I wasn't able to meet him and follow up what he said, but he stood up and said that he had, over about 15 years, walked himself free of all his symptoms of Parkinson's, which is quite an interesting claim. He looked pretty fit, and he was in his, I would guess, in his late 70s. But that was his claim, that he'd actually walked his way out of the symptoms.
There's a person who's got a blog on Facebook called Dr. Gary Sharp, who just is promoting exactly that.
Right.
The visualization of movement. And he videos himself, and it's absolutely incredible to watch. You can see him when he's frozen, and when he does his visualizations walking. He uses balloons and rings and all sorts of things in music, and talking and poetry reading, and he writes songs and things, and it really is fascinating. And just
yes,
he sounds like he is trying to walk himself away from Parkinson's.
Right. So
the person you refer to is in chapter 2 of Deutsch's book it's something like, it's his second book, it's about how the brain heals itself.
right.
So that's very well written up, and I can highly recommend the book by Deutsch.
So, as you can see, we're scratching the surface of something very, very big and well worth investigating here. One of the things that Roger Barker was really emphasizing was beware of any new advertised stem cell trials, especially if you're being asked to pay to join it. There are some dodgy promotions going out there which are making great promises to people and inviting them to pay considerable sums of money to join the program in the hope that this will provide them with a cure. It does carry quite clearly quite considerable risks. So this really takes us forward into the genetics area. And the question, can we discover genetic risk factors and head off the potential for Parkinson's disease? Of course, you immediately hit another whole order of complexity. And this was what made a huge impression on me, was just, one keeps on plunging into enormously complex graphics that these wonderful people prepare. Now they understand them, but I'm blessed if I do. And I just like the smiling face of Robert Nussbaum there, because he clearly he's into this stuff and with tremendous success. But the point about this is that if you take a population, a kindred, a family kindred, that have exhibited several people who, among their relatives who've prone to Parkinson's disease, if you plot all the genetic factors there and how they show up, and you find that some people, although they carry the gene, are less prone to Parkinson's, and other people who also carry the gene are highly prone to it. And what is going on here? What's the genetic sort of process that's happening? And how can you, from that, possibly predict people who are susceptible to Parkinson's and then take preventive measures right at the early start of the process? That's clearly a really important line for research, but it is again fraught with all sorts of complexities. And one of the things, of course, is what do you tell people themselves? How would I, as somebody who'd been identified as having that genetic risk, that genetic propensity, then deal with that information? So this was an important presentation by Alice Lazzarini, and it leads into looking at what is the this maps out the risk factors. So up here, if I can point that, oh, is this working? Got my finger at, yeah. These are the genetic, the genes which provide quite a high risk of causality, but which are very rare in the population. And these are the ones that have some risk, but are quite common in the, or more common in the population. But here you've got an intermediate group, which are both high in risk, but intermediate in frequency. And do those offer some route for looking at populations and testing out, and then some prevent, you know, could that lead to preventive activity, preventive therapy? So here's a worldwide spread of the LRRK2 gene, and with its strong risk factor, and that shows. If I then go on to looking at, but however, when you then take genetics and environment into action, it really gets incredibly complicated. That's just an illustration of that kind of complexity, and there's no point in going through the detail of the slide because I can't do it. Yeah.
So this means that there are these particular areas with
yes, which have much higher propensity. Yeah. And there are also areas with much less less effective health systems, actually.
So the circles are all the areas they all have high general risk.
That
it's not the 30%, is that the red bit?
That's I think that's the estimated penetration of those genes among the population. Yeah, it's pretty high. But that doesn't mean that they're all going to get Parkinson's, because you've got this really subtle, complex things of what is in each individual the risk factor, the combination of risk factors or protective factors, and then what in their environment and experience. Actually, so it doesn't mean, it's not a predictive map. That is, it just says here is a map of where the genes are. Yeah. Okay, and this isn't predictive either. This is just illustrative of the different things that affect people and can, in certain combinations, bring out Parkinson's. And then this is if you get into environmental self-reporting. The previous slide was sort of factors that, if you like, go on inside people, and these are factors outside people, self-reported effects. And then is there some protection or reverse causation from other things, and so on. So that just, again, it's an illustration of the complexity. Yes, gout. That was the thing.
We'll be looking at all the bits on that side of it later on. We'll be looking at those areas later on, and all the things that, I mean, they say that genetics loads the gun and environment pulls the trigger. So we'll be looking at how you can help to not pull the trigger. But gout is because of the uric acid, because uric acid is an anti-inflammatory. So if you've got gout, then you're less likely to get Parkinson's.
And obviously the tantalizing thing is reverse causation. Is this, what might actually help and protect? Again, it's highly individual. We were told right at the start, what the medics say to themselves is, if you've met a person with Parkinson's, you've met a person with Parkinson's. You have not met a whole cohort. It's so individual. a lot of work being done on pesticides, because California happens to have not only a very high rate of pesticide application, colossal rate actually. I was amazed at the intensity with which the Californians are applying pesticides, but they also have a very strict recording system, which means that you can plot the incidence of pesticide application around particular homes and people living in those homes, and map out, and if they show up with Parkinson's, you can actually plot the times and the whole thing. You can put it together, put the whole pattern together as to whether this or that application had a causative effect or profound influence on the emergence of their Parkinson's, again with their genetic patterns and so on. So there's a huge amount of work going on in this area, but what was clear to me was that, I've always thought this, but this brought this out to me very, very clearly, that pesticides are utterly inimical to human human thriving, human growth, human living. And it starts, again, because life is so interconnected genetically, that bees and wasps and little insects all have DNA just as we do. And so if you're killing one kind of life, how do you avoid killing other kinds of life? That seems to be something fundamental. I'm just amazed at the alacrity with which people use pesticides. So that's the great aim of all of this work, and you can see this pesticide application stuff, and putting that together with the genetics and seeing what kind of understanding comes out of that. And then there's yet more potential with epigenetics. And I don't fully understand epigenetics, and I think Ken will have a much better handle on it than I do. But basically, it gets into this business of what is the causative pattern in populations that can give rise to disease manifestation. And now, with technology and computing power, that really becomes, and information gathering, that becomes a possibility. You can see, I won't try to explain all the details of it. Then there's the question of what happens with the information. Because there's now, this is from Alzheimer's, but there's now a sense that people who are told about their susceptibility or otherwise to a condition, it feeds back into how they perform in things like memory tests and so on. And people who are oblivious to whether they're susceptible or not are tending to do better than those who are informed with this knowledge. And so you wonder what goes on inside us. How do we script ourselves? What are the stories we tell ourselves, and how does that affect our responses to these conditions?
It's an issue for me personally, because I'm part of a study which is looking at whether or not there is a genetic component to my own Parkinson's. Now, looking at this information, I'd always thought, well, perhaps my children would want to know. But looking at this information, it's raising the question that if I do tell them, then that's more likely to trigger their Parkinson's if they have a susceptibility for it. And it's a real moral dilemma here. And the other thing is that because Parkinson's has such a huge placebo effect component, it also has what I've discovered is called the nocebo effect, the hex, the medical hex. So as a result of this information, I'm thinking that if there is a genetic component to my Parkinson's, that I shouldn't tell my children, because that would be more likely to trigger theirs. So there's a very useful piece of work, this. Is anyone else part of a a genetic study?
It takes us into this whole mind process. What goes on in our minds, and how do we, how do we tell ourselves? Because there's a huge amount that says that with mindfulness we can improve our well-being enormously.
Well, it's back to what Linda was saying about the visualization, isn't it?
Yeah.
So
yeah.
I think we should crack on.
Yes, we should. Yeah. This business about taking genetics personally, what can you do and what will it do to you? So I think we've covered that. I put this in just to show the further complexity. This is about could you develop a vaccine against PD? And one of these slides came up in a presentation. She said, rather playfully actually, this is the kind of thing you get into. So I put it up because it just shows how anybody actually devises a slide, has the understanding to devise a slide like that, I don't know. But it just shows this is, if you're getting into prevention, this is the kind of complexity that goes. This is to do with experimenting with mice, I think, and probably with pigs too, as to what if you induce Parkinson's in these animals, then can help prevent it spreading or even restore better function. And lastly, the whole business of accelerating research. This was a fascinating presentation about automated disease modeling. So what this wonderful man, Stephen Finkbeiner, was doing, he'd got an automated laboratory which was generating vast numbers of induced stem cells and then experimenting with different varieties of inducing Parkinsonism in them, and then experimenting with huge varieties of potential cures, of potential medicines. And so what this meant was that he could accelerate the process of both cell selection, cell testing, and mapping out results at an extraordinary rate. So it was really very, very impressive, that, and how he could test for various drug potencies and so on, and that I think offers again a big potential advance in the process, because he said that if you can automate the whole retraining process for iPS cells, then you can accelerate the testing, and if you can increase the range of those, so you don't have, because you can't have researchers looking at them 24 hours a day, as you ideally would want. So that whole process can accelerate. Okay, and then this is the potential that comes from that, modeling the disease and modeling treatments for it. So that was kind of, and the takeaway messages from that: patient-derived cells offer exciting new opportunities. Human brain cells from PD patients exhibit significant changes in survival and other endpoints, and using these patient cells to understand PD and find therapeutic targets and therapies is the key thing. Okay. And the whole thing about teamwork, of course. This came through the Congress the whole time. The collaborations, elaborate collaborations, right through the system. It's time for a stretch. That's as far as we get on this section. And I do thank you for your patience, and I hope that's been informative, sufficiently informative, and apologies for my incompetent scientific knowledge.
And thought, I'm not daft, I'm the one that decided that David was much better placed to do this, so I skipped on to something much easier. Linda, you had a question.
If you can give us a very interesting talk, I think about a year ago, about the research that's being done with people on specific kinds of medicines, like for hypertension or diabetes, that showed that these people have a lower incidence of Parkinson's disease. Was there anything more about that?
I think Ken's probably best asked to answer that.
Two things. There's a general area which is becoming very important and fashionable, which is called drug repurposing. That's an attempt to identify beneficial effects for Parkinson's from drugs that are already approved for other conditions. In particular, there's a trial in University College London of a diabetes drug called exenatide, and the results from that are just coming out now. There's also an attempt to find out from patients themselves whether they've come across anything that they think improves their Parkinson's conditions. The Parkinson's UK Research Support Network development team, on which I sat until recently, has put together a project which involves sending out questionnaires to people to ask them whether they'd noticed particular beneficial effects from taking other medicines of any sort, and to correlate that across the country. So that there is an attempt to identify whether there are factors that people are already using that might help people with Parkinson's.
Well,
I'm interested in this discussion on genetics. I've got Parkinson's and I've got a twin brother. Does anyone know if there's been any studies with twins and what are the likely chances of him having Parkinson's as well?
I'm not aware of any particular studies on that. That doesn't mean they're not happening, because there's a huge medical interest in twins, and there's a lot of work that goes on in all kinds of fields to do with twins.
We didn't see anything at all in the Congress about
We didn't see
that.
anything in the Congress, but clearly that would fit into the whole family cohort genetic mapping process.
It's the sort of question that you could ask Thilo this afternoon, Thilo Kunith, because he may have more insight. There's one at the back there first.
I'm a relative newcomer to all this, and this may seem rather a daft question, but it seems to me everything comes back on the medication front to levodopa. Is it still the only main medication available?
There are two, there's levodopa and there's dopamine agonists. Those are the two main ones. But, I mean, levodopa was discovered 50 years ago next year, and it's still really the only kid on the block, apart from the agonists. Yeah.
Thank you.
Linda, you heard one.
Just when you touched on a lady using a nicotine vape, this talking about the delivery of the drugs. Was she using the nicotine, or was there something else she was using via the vape?
No,
to
nicotine. Yes.
Help the dyskinesia.
It helped her with her dyskinesia. Yes. So we'll look at the role of nicotine in helping symptoms.
Just to add my two pennyworth, nicotine has been known for quite a long time to be supportive of resistance to the development of Parkinson's. So people can, if they wish, increase their or decrease their risk of Parkinson's by chain smoking, but most medics would not recommend it as a suitable treatment. Likewise, there's evidence that large doses of caffeine are neuroprotective, so if you've been a heavy coffee drinker all your life, your risk of getting Parkinson's is reduced.